present Kentaro. Kentaro, hold on. Kentaro did his medical studies in Japan, in Tokyo, and then he moved to the US to do his pediatric residency in Oakland University, and then he moved to Washington University, St. Louis to do a prolific postdoc at Marco Corona's lab where he studied intraepithelial cells. And now he is an assistant professor at the University of Colorado. And I'm welcome, Kentaro. I'm happy that you accept our invitation. And before we start, I would like to ask here in the audience a lot of scientists or trainees who are outside the US, any advice to people who want to do research in the US or in another country that is not theirs?
Yep. So when I first came to the US, I really wanted to do science, but I went to medical school. I didn't have any experiences, but I keep sending the applications, and most of my applications were ignored. But the group of people who showed me invited me working as a postdoc. And as long as you have a passions and you what you really want to work on, there's always a group of people who want to support your passions. So be passionate about your science, love the science, and showing how you love and science and why you want to work with those people. And there's always people who will support you.
Thank you very much, Kentaro, and I give you the floor.
Okay. So thank you for inviting me to the seminar today. My name is Kentaro Yomogida. I'm a pediatric rheumatologist at the University of Colorado. Um, I did a postdoc at the Dr. Merkel Coroner's lab, Washington University, and I started my lab last summer at the current institute. When I was in Marco's lab, I have been studying the mucosal immunology because as a rheumatologist, I have a strong interest in how autoimmune disease is developed, and the mucosal area is a location immunotolerance is broken. And we have been looking at a specialized group of lymphocytes called intraepithelial lymphocytes. So today I want to talk about how activations and the function of IELs are regulated at the transcription level. Our bodies are constantly exposed to trillions of microbes, and the gut hosts the largest amount of bacteria inside the bodies. Most of the commensal microbes are harmless, but because of this unique anatomical location, we have multiple layers of protection in intestine. First layer of the protection is mucus layer. Mucus is a thick liquid that is filled with antimicrobial peptides, and this functions as the first layer of protection to quench the bacterial activities. Second layer of the protection is epithelial cell layer. One layer of epithelial cells cover entire intestine. And this epithelial cell layer provides a physical barrier between external environment inside the bodies. And beneath the epithelial cells are connective tissues termed lamina propria. And diverse group of lymphocytes reside in the lamina propria, constantly monitoring infection or potential danger signs. There are a group of cells termed intraepithelial lymphocytes or IELs. These cells are residing and mobilizing along the epithelial cells, constantly checking the infections and or any damaged tissues. And there's a one IEL in every 10 epithelial cells. Because of a large surface area of the gut, IELs are the largest group of lymphocytes inside the bodies. There are some differences between mouse and human IELs, but I want to talk about the subset of IEL in the mouse because my project is focusing on the mouse models. Majority of IELs are T cells. Close to 80 to 90% of total IELs are T cells. Then they can classify it into conventional, unconventional IELs. Some people call induced or natural IEL, but we call it here conventional and unconventional IELs. Conventional IELs are either CD4 T cells or CD8 T cells. Small group of CD4 T cells acquire cytotoxicity, and sometimes they are termed as cytotoxic CD4 T cells, or because they simultaneously expressing CD4 and CD8 alpha alpha chain. Sometimes it's called double positive IEL. And unconventional IELs, they are either expressing gamma delta T cell receptor or they are expressing alpha beta T cell receptor, but they don't express CD8 alpha beta or CD4 chain, but they express CD8 alpha alpha. And they are often termed CD8 alpha alpha IELs, or because of lack of expression of CD8 alpha beta or CD4, they are called double negative IEL. Today, I'm just terming this cell type as DNIEL, meaning double negative IELs. And unconventional IELs are the predominant form of IEL. 60 to 80% of total T cell in the intraepithelial layers are unconventional IELs. Primary role of IELs are considered to be protection from infection. Many study has shown that specific bacteria or viral infections are protected by the IELs. And but IEL also producing the some of the growth factors. So these growth factors promoting tissue repair when the tissue get injured. And overactivation of any immune cells can harm the host, and the IELs are not the exceptions. Overactivation of IELs are indicated in certain gut inflammatory conditions. For example, the celiac disease, IEL activation seems to be contributing to the atrophy of the villi. In the certain IBD patients, some subset of IELs get more prominent, suggesting IEL overactivation may be part of the pathogenesis of celiac and Crohn's disease. How IEL activations are regulated. And activation mechanism IEL has been studied intensively, and I think we can summarize into the 3 points. First, activating receptor and inhibitor receptor. So IEL expressing multiple inhibitory receptors or activating receptor, where the signaling from the activations or inhibition is the stronger. That balance determines how IEL behave. The second mechanism is activating cytokines. Some cytokines like IL-12, IL-15 are known to activating IEL. And because IEL is constantly exposed to the external environment, some environmental stimuli, such as HL ligands, are known to activating IELs too. But we do not understand what types of transcription program regulating IEL activation. And we start looking at the molecule called Aiolos. Aiolos is a transcription factor and one of 5 IKGF family member. IKGF family members have 5 proteins, and they have similar sequencing and expression profiles. And Aiolos is number 3, and this is predominantly expressed by the lymphocytes or some small subset of granulocytes. This molecule was identified 30 years ago, but we still do not understand what exactly they are doing in the immune system. The things we know about is they seems to be important for maturation of NK cells or activation of B cell and ILC2, or migration of eosinophil. But we don't know what they're doing in the gut immune system. The other reason we are interested in the Aiolos was polymorphism of this gene associated with many autoimmune conditions like lupus or IBD. So we thought Aiolos may be important molecule regulating gut immunity, and we started looking at its function in IEL activities. So today, I want to focusing on the function of Aiolos in the unconventional IEL, which means gamma, delta, and double negative IEL, because these two are the subsets that the predominant cell types of the IEL. So first, I looked at the expression patterns of Aiolos in the unconventional IELs. This is very simple experiment, just harvesting the IEL from the mice and stain with the Aiolos. Both gamma, delta, double negative IL express very high level of Aiolos. So to understand the function of Aiolos in the unconventional IELs, Um, we got the Aiolos knockout cells mice, and we profile the phenotypes of the unconventional IEL. And we have noticed unconventional IELs express very high level of NK receptor. NK 1.1 is a universal NK receptor in the black 6 mice. Unconventional IEL from the Aiolos knockout mice express a very high level of NK 1.1. So something seems to be going on. So to understand what Aiolos is doing, we did bulk RNA sequencing. We isolated gamma delta or double negative IEL from wild type or Aiolos knockout mice and compared gene expression patterns. And we have noticed Aiolos deficient IELs upregulating granzymes, interferon gamma, and NK receptors. What does this mean? Unconventional IELs, both gamma delta and double negative IEL, they are known to producing granzyme gamma interferon and upregulate expressing NK receptor when they are activating. So these patterns of the gene expression indicating Aiolos deficient IELs are more activated. In other words, Aiolos is a potent repressor of unconventional IEL activations. Now, Aiolos deficient IELs are more activating, so we are trying to looking at whether Aiolos deficient IEL alters the host response to pathological conditions. And we use a chemically induced colitis model. DSS is a chemical that are toxic to the epithelial cell in the gut. So when we give a DSS in the drinking water to the mice, epithelial cells get damaged and causing the colitis. So we put the DSS in the drinking water and provided it to the wild type and Aiolos knockout mice. And we looked at several parameters that indicating the colitis. First parameter, we looked at the blood in the stool. Both wild type and knockout mice, they eventually start bleeding in the stool because it is damaging the epithelial cells. But Aiolos deficient mice, they start bleeding at the earlier time point. The other thing, we looked at length in the colon. Colon length is one of the parameters of the colitis. When mouse has a severe colitis, colon lengths get shortened. And compared to the control mice, Aiolos deficient mice has shorter colon lengths, indicating Aiolos deficient mice has more severe colitis. So Aiolos deficient IELs are overactivating, and this overactivation deteriorating the chemically induced colitis. So Aiolos is a restraining unconventional IEL activation, but the question came to our mind is whether unconventional IELs are homogeneous group of cells and the Aiolos affecting the entire activation of IELs or not. Or actually IELs are heterogeneous group of cells and the Aiolos affecting a specific population in among the unconventional IELs or not. To answer these questions, we did a single-cell RNA sequencing. First, we did not know how heterogeneous unconventional IELs are. So I want to show the heterogeneity of gamma, delta, and double negative IELs. So this data is a combination of a single-cell data of gamma, delta, double negative IEL from the wild type mice. I'm not going to talk about the detail of each cluster, but I want to summarize the three important points. First, gamma, delta, and double negative IELs, they are indistinguishable on the single-cell RNA sequencing. Up to this point, I have been discussing gamma, delta, and double negative IELs are different cell type because one cell is expressing gamma, delta T cell receptor, the other subset expressing alpha, beta T cell receptors. However, at the single cell level, they are undistinguishable. The other thing we have noticed was there is a stimuli to effector transcription program. So stimuli T cells, either CD4 or CD8 T cells, they express high level TCF7, protein name is TCF1. And when they got activated and become effector T cells, they down-regulating TCF7, they up-regulating PRDM1. This protein name is BLIMP1. we see exactly the same transcription program in the IEL. The third, effector IEL, these TCF7 negative population, they are classified into two groups based on the interleukin-15 receptor expression. One group expressing high level of interleukin-15 receptor, the other one expressing low level. And these two groups producing a different type of cytokines. So the IELs are more heterogeneous than what we thought. Then I want to go back to the original question we tried to answer, whether Aiolos is affecting the one group or they're affecting, Aiolos is affecting entire groups of the unconventional IELs or not. So we compare the single-cell RNA sequencing between wild type and the Aiolos knockout unconventional cells. Comparing both groups, both groups have TCF1 positive stem-like IEL populations, and both groups have interleukin receptor low effector IELs. However, in the ISO knockout cells, interleukin-15 receptor high populations are completely gone and forming a new cluster. This is suggesting interleukin-15 receptor high population are heavily affected by the Aiolos, and the Aiolos reprogramming transcription program of interleukin-15 receptor high effector IELs. So this is the computational data. So we try to think whether these data can be validated on the actual mouse cells. First thing we looked at is whether we can separating the gamma delta double negative IEL based on the expression of TCF7 and the interleukin-15 receptors. And indeed, gamma delta and double negative IEL can be classified into three groups based on the expression of TCF1 and the interleukin-15 receptors. The then question was whether IO is predominantly affecting the only one group compared to the other groups or not. So we looked at the three groups of NK receptor expression in the wild type and knockout cells. And compared to the TCF1 positive population, or compared to TCF1 negative but the interleukin-15 receptor low populations, interleukin-15 receptor high population express a very high level of NK receptor. So this is indicating interleukin-15 receptor high population is more affected in the absence of Aiolos. But why only this population got affected compared to the other populations? So we thought one of the key function and key molecule is interleukin-15 because IL-15 is one of the critical molecules supporting survival and the activation of unconventional IEL. We thought IELs may be controlling the responsiveness to interleukin-15. So to assess the sensitivity to the IL-15, we did an in vitro assay. We isolated IELs from wild type and knockout cells, and we cultured in the different concentration of IL-15 to look at the responsiveness to the IL-15. When we culture low concentration of IL-15, wild type cells do not express any NK receptor, and knockout cells express very little NK receptors. But when we culture high concentration of IL-15, wild type cells start expressing high level of NK receptor, but this upregulation is more pronounced in the knockout cells. So these data are suggesting Aiolos is controlling the sensitivity and responsiveness to IL-15. And that is why IL-15 receptor high population is more affected in the absence of Aiolos. The last question we tried to answer was how Aiolos is restraining IL-15 stimulations. To answer this question, we need to look at the IL-15 signaling and signal transduction pathway. When IL-15 receptor recognize IL-15, they use the JAK-STAT signaling, in particular STAT5. And this activating interleukin-15 receptor phosphorylated STAT5 and inducing the various activating molecules. Because STAT5 is transcription activator and the Aiolos is a transcription repressor, we thought they may bind in the close proximity, maybe counteracting each other's functions. To understand where Aiolos and STAT5 binding to, we performed cut and run sequencing. Cut and run sequencing are new version of a kind of chip sequence. It's a different technique, but a similar technique. And we do not need a large input like a chip sequencing. And the background was the small is less background. So we use the cut and run sequencing to understand where Aiolos binding to. And we use the STAT5 publicly available dataset. First, I want to show the example plot of the Aiolos and STAT5 binding region. This is a snapshot in the loci of granzyme B. I also start the file clearly binding the very close proximity in the very specific loci. Then how about on global chromatin binding locus? So we looked at how many regions ions bind to and how many DNA regions STAT5 binding to, and we compare number of overlapping binding regions. And in the Aiolos binding region, we observe around 5,000 to 7,000 region in the double negative gamma delta IELs. STAT5 around 11,000. And among those binding regions, about 1/3 to 1/4 of the Aiolos binding region are overlapped with STAT5. And these overlapping regions are in the close proximity of IEL effector genes such as NK receptor, chemokine, gamma, interferon, and granzymes. So these data are suggesting likely Aiolos bind close proximity to STAT5, and because they're binding close regions, they are unblocking each other's functions. Summary of my talk is unconventional IELs expressing high level of Aiolos, and Aiolos potently regulating the function of IEL. And Aiolos deficient IELs are more activating. That is why they're deteriorating the chemically induced colitis. Single-cell RNA sequencing data shows unconventional IELs can be classified into 3 groups based on expression of IL-15 and TCF1. And Aiolos specifically impacting the transcription of the interleukin-15 receptor high effector IELs. And Aiolos controlling the responding to the IL-15. So Aiolos deficient cells are more responsive to IL-15. How they accomplish it? Because STAT5 Aiolos binding to the close proximity, they're counteracting each other's function. So this is a collaborative work, and I couldn't complete this project without help from the Corona Lab and Washington University. And in particular, I want to say thank you to the three senior faculty member in the Marco Lab, Marco, Marina, and Susan. And and also the chemical induced colitis model, I got help from the Tihana. We also collaborated with the group in Ohio State University, Jean Olsen and Patrick Collins. They helped us to understand the epigenetic studies. And I started in my lab. I'm still working by myself. I'm looking for somebody who is interested in joining my lab. That's all I have today. I'm happy to take any questions.
Thank you very much, Kentaro, for such a great presentation. So if anyone in the audience have any questions, please raise your hand or type it in the chat. Meanwhile, Kentaro will just ask, I have particular 2 questions.
Yep.
What is the factor or the signal that induce the expression of Aiolos?
Yeah. So the Aiolos is known to be induced by the TGF-beta and the interleukin-6 signaling. And when I looked at the interleukin-12 and the type 1 interferon, like interferon beta reducing the expressions. And when I did a poly-IC stimulations, so the poly-IC is a ligand to Torak receptor 3 and inducing a very high level type 1 interferon or systemic inflammation. It's also bring down the expression of the Aiolos expression. So Aiolos seems to be reduced by the inflammatory signaling, but induced by the IL-6. It's also inflammatory signaling. I don't know why. And also TGF-beta.
So if it seems that an anti-inflammatory signal is the factor that induce Aiolos, then My second question would be, does this factor is affected in the absence of microbiome? Like how is the intraepithelial cells, uh, their activation state in the absence of microbiome, for example?
Um, so the so the if IEL activations, so in the absence of Aiolos, IEL composition get altered quite significantly. And in the absence of Aiolos, they do not have a constant signal from the microbiome, so that they do not have activations. But IEL activation is tightly regulated at the steady states, even in the presence of Aiolos. We often call it resting, but activating a resting status, meaning they look like a activating, but they do not constitutively producing the effector cytokine or cytotoxic enzymes. So in the presence of microbiome, they do not always get activated.
Thank you very much, Kentaro. I don't know if there's any more questions in the chat box. I just have a briefly question that's really like basic. I remember that in immunology books they said that after born you have the very first waves of gamma delta T cells that infiltrate the tissues. Does this happen with the same with the intraepithelial cells? You have different waves of intraepithelial cells after birth.
So
Yeah,
yeah,
yeah.
that's very good questions. We still do not understand the ontology of the IELs quite significantly yet. And then probably gamma delta have some in general, the CD8 alpha alpha IELs are known are probably coming from the thymus, but there may be some extra thymic pathway. And the gamma delta IEL, there's some controversy. Some are coming from the thymus, and some are probably coming from the extrathymic, maybe fetal liver. But we still do not understand what kind of wave there exist.
Thank you, Kentaro. I think there's a question in the chat, and Julian Grandveld asks, double negative or gamma delta intraepithelial cells, which show more stem-like features, especially when comparing to the TCF7 expression. I think
Uh,
if you also if you can read the question in the chat, my audio is some
for
kind
the
of
most
blurred.
features, especially on the comparing TCF7 expression. Yeah, that's very good questions. So this seems to be having some different functions, and the cytokine production seems to be quite different. So in particular, they're expressing XCL1. So they seems to be probably recruiting the dendritic cells, and in terms of that, they have some form of the stem-like features, but they probably behaving a little bit different from the so-called but we still do not understand whether they are self-proliferating, self-regenerating or not, those kind of stem-like features or not. We haven't done that very detailed study of the stem-like TCF1 positive populations yet.
Okay. There's another question. She
Oh,
said,
okay.
yeah. Mhm.
Yeah, that's very good questions. So the Aiolos, whether the question was whether IKJ family member is forming a homodimer and heterodimers, and whether Aiolos does the homodimer forming homodimer or heterodimers or not. So the only thing we know about is Aiolos forming a homodimer, or they can form a heterodimer with Icarus. So in the absence of Aiolos, there may be some compensatory mechanism with the other Icarus IKJ family member. For example, IKGF1, Icarus may be compensating for it, or maybe Helios, IKGF2 may be compensating for it. We haven't made a double knockout mice, so we do not know yet. But there's a recent paper showing IKGF1 and the 3 knockout mice. So Icarus IOS double knockout mice has a more significant phenotype compared to the 1 knockout mice. So I'm pretty sure there must be some compensatory mechanisms.
Mm. Thank you very much, Kentaro. If they ask where's your lab, I think your lab is in the University of Colorado, and I will put in the chat the information so you could contact Kentaro.
Thank you.
Thank you. So